Thanks so much for the invitation. My name is Ricardo Dolmetsch. I'm the global head of neuroscience at Novartis, which is one of the largest pharmaceutical companies in the world. And for the last 5 or 6 years, we have made major investments in stem cell technology, both for therapeutic purposes as well as to model disease. I want to spend a few minutes telling you about why we believe in the technology, the kinds of things that we are doing, and how we're doing them before I cede the podium to my fellow presenters. Drug discovery is largely about developing preclinical assays that model the human disease. And in neuroscience, we have been, I should say, not terribly successful at developing animal models for many of the diseases that we're tackling. And the reason we know we've been unsuccessful is because we are facing about 2 decades worth of failures in neuroscience. There have been, I think, some recent successes, but broadly speaking, we have not really changed or moved the needle for some of the worst diseases that affect the brain. And as we were thinking about how to do things, what we could do differently, we decided that one of the things we had to do was come up with better preclinical models. So we took a page from our colleagues in oncology, and one of the things that the oncologists have learned is that it is very useful to have human biopsies. And in oncology, you can take a biopsy of a tumor and you can study it in the lab, or you can introduce it into a mouse, and you can treat that biopsy sort of as if it was a tumor in a human. Unfortunately, we have not been able to do that in the nervous system because most people are unwilling to give up a little piece of brain to model a disease. And so as we were thinking about how to do that, we thought, well, if we can't get a biopsy, maybe we should make one. So the question is, can you really make a biopsy? And the answer is yes. You can take skin cells or blood cells, and you can reprogram them to make stem cells, and then you can take those stem cells and convert them into neurons. And you can convert them into neurons that grow on a sort of 2-dimensional surface, or you can create these sort of mini brains, these organoids. And in both cases, you can then see whether you can detect any cellular abnormalities, any phenotypes that are associated with the disease. If you identify phenotypes, then you can use those phenotypes as a way of trying to develop new medicines. And so this is something that we've been doing really over the last 5 years. Now, we've had to solve a bunch of issues because the technology is actually pretty new. So among the issues that we've had to solve are, for example, how to make the process efficient, right? So when I first started working on this as a professor at Stanford, it took a postdoc many months to differentiate just a small number of cell lines, and it was heavily dependent on the skill of that individual. This obviously is not plausible if you need to study hundreds or thousands of drugs, and so we had to develop automated ways of differentiating the cells. We had to develop ways of making different kinds of neurons. We had to develop more sophisticated ways of identifying phenotypes. And over the past 5 years, we've been, I would say, modestly successful to the point where every neuroscience drug discovery project now has a stem cell component. Now, we also recognize that stem cells and stem cell-derived neurons are not enough, that we continue to need animals, especially for pharmacodynamics, which is sort of the distribution of drugs in animals, and in humans, and for safety studies. But in a few cases, we're just looking at the efficacy of the drug in a cellular model, especially in cases where the cellular phenotype is very clear and seems to recapitulate the phenotype of the patients. So there are a couple of examples where we've been quite successful. One of the examples is in spinal muscular atrophy, where we have used patient-derived cells to restore the expression of a gene that is missing in these children. We've developed a small molecule. We've taken the molecule all the way to humans, and this is a molecule that is now in clinical development, and we have exciting clinical results. We have worked on a series of other neurodevelopmental diseases. We work on Dravet syndrome. which is an early onset epilepsy. Again, we've made the right kinds of inhibitory neurons and developed small molecules for them. And we continue to work at developing the technology further, making better organoids, making things more reproducible, and trying to sort of move the field forward, because we think that there are huge advantages to having a human model to develop medicines. So that is kind of the Novartis story. And now I'd like to just sort of ask some of my fellow panelists. So I'm just going to introduce the panelists here. So, and I apologize if I mispronounce somebody's name. So Hynek Wichterle is from Columbia University. He's an expert in the development of neurons in the spinal cord, and he's made major contributions to understanding how they're generated. Ed Stanley comes from the Murdoch Children's Research Institute, where he's worked on the differentiation of stem cells into endodermal lineages like kidney and blood. And Joe Wu is the director of the Cardiovascular Institute at Stanford, and he's made major contributions in using stem cells to generate cardiomyocytes and understand phenotypes associated with various kinds of cardiomyopathies. So, Hynek.
Thank you, Ricardo. So my name is Hynek Wichterle. I'm a co-director of the Center for Motor Neuron Biology and Disease at Columbia University. And actually, this was a really excellent introduction to the questions that I'll be discussing in my presentation. So you can see here that I highlighted some of the points that Ricardo already mentioned, that neurodegenerative diseases, as we are getting more and more successful in treating diseases such as cancer, will become a major issue, as the prediction is that the population over age 65 will double within the next 25 years. And unfortunately, currently, there are no cures for many of the degenerative conditions of the central nervous system. And so, obviously, the development of stem cell technologies was heralded as a major step towards not only modeling diseases of the CNS that, as Ricardo mentioned, we cannot really reach into the patient's brain and take a biopsy to study the cells in culture, but also for discovery of new potential treatments. But this is not a straightforward path. And so here on this slide, I would like to highlight a few of the challenges that I find associated with successful modeling of neurodegenerative diseases. And my laboratory is primarily interested in one disease, which is amyotrophic lateral sclerosis, a disease caused by degeneration of motor neurons. So one of the issues is that the degenerative diseases do not affect all the brain cells equally. There are some small subsets of cells that are highly susceptible to degeneration, and we need to therefore model the diseases in the appropriate cell type. So for that, we need to understand how different cell neuronal identity is specified in the embryo, and we need to be able to recapitulate the same process in culture dish in vitro. And so that's the first issue, the neuronal diversity that we have to that we are challenged with. The second problem, which I believe is even more concerning or harder, is the fact that these diseases are caused by mutations in genes that are expressed from the embryonic age. But for most of the lifespan, these mutations are latent. They do not actually exhibit any phenotype. But then something happens during aging when these mutations start causing neuronal loss and eventually the phenotypes that lead to a patient's death. And so there's very latent or even subtle phenotypes that are caused by these mutations highlight how difficult it will be when we try to model the same pathologies in culture dish. Next issue, which I would like to highlight, is the fact that many of us who work with pluripotent stem cells in culture know that each cell line is slightly different from the next cell line. And so these subtle differences, or these cell line to cell line differences, and also cell culture to cell culture differences might contribute sort of noisy readout of these assays, which will make it even more challenging to detect these subtle phenotypes that I discussed that are associated with neurodegenerative diseases. And finally, because the disease starts in patients, but then the progression can extend over several years, it's often difficult to actually even distinguish which of the changes that we see in patient tissue are truly pathological, causing neuronal death, and which of the changes are actually homeostatic or protective responses of the tissue that are trying to prevent the neuronal loss. And so how do we try to circumnavigate some of these challenges? So many years ago, we developed very efficient ways how we can model motor neuron development from either embryonic stem cells or induced pluripotent stem cells. We developed now very efficient ways how we can use the signaling molecules that normally control motor neuron specification in the developing embryo, patterning signals, and basically instruct embryonic stem cells to differentiate very efficiently into motor neurons that we can then actually implant into developing chick embryos and show that these motor neurons are functionally equivalent to normal embryonic motor neurons formed in the developing chicken. The other way, we found way to reprogram actually embryonic stem cells very efficiently into motor neurons using combinations of transcription factors. And that now provides a unique opportunity where we have cultures that are nearly pure, populated only with motor neurons. So that gives us unprecedented access to this one particular cell type that is lost in ALS. Now, how we went about to try to model the subtle features of the disease, the subtle phenotypes that might affect motor neuron survival in these cell-based models. The way we wanted to avoid some of the noisiness of cell culture to cell culture differences was that we actually decided to co-culture in the same well in Petri dishes both the control cells that were marked with red fluorescent protein and ALS motor neurons marked with green fluorescent proteins. And in this way, we can really compare side by side the survival or ability to cope with different stressors of these two types of neurons. So now, with this approach where we can generate this two-color system, we can plate motor neurons in multi-well plates. In this case, we are using 96 well plates. And then the first thing was sort of counterintuitively, we did not look for drugs that would promote motor neuron survival, but initially we wanted to find drugs that would actually accelerate the disease pathology. So we were looking for stressors that would selectively kill the ALS, the green motor neurons, and left the red motor neurons surviving. This assay brought us to actually a series of stressor molecules, some of which induce protein misfolding in these ALS motor neurons. And you can see here an example of one of these stressors when under control conditions you see beautiful motor neurons with neurites, and under stress conditions there is a remarkable degeneration of these motor neurons. Now we could revert the screen and look actually for drugs that would prevent the degeneration of motor neurons that were exposed to these stressors. And this reversal screen actually led us to identification of several interesting molecules that completely prevent motor neuron degeneration under these stressful conditions. And this is quite remarkable because we can now effectively treat this very simplified ALS model in a dish. Yet it is a very long distance from having a drug that works in a dish going to a drug that works in patients. And so what we did next was that we performed a series of chemical optimizations. We generated more than 80 different analogs of this drug. And now we have compounds that actually have very favorable pharmacokinetic properties, so we can go in mouse and test these drugs in animal models of ALS. So finally, I just wanted to touch on two remaining challenges that I believe really slow down our progress in using stem cell-based models of diseases to discover new treatments. So one of them is that what I showed you was basically single pathology modeling. So we modeled the protein misfolding in motor neurons But we know also that ALS is not caused by a single pathological insult in motor neurons. There is many different things that happen in the aging motor neurons that we are probably not modeling in the system. And so I believe that going into future, we will have to find a way how to really basically accelerate this natural or aging process in a dish to try to recapitulate many aspects of the aging motor neurons. And second issue that I believe we did not really circumvent is the fact that there is a lot of variability between individual lines. And so for that, I think that the next reincarnation of these screens should be performed with, by multiplexing many different control and disease lines in the same well. As I showed you, we can multiplex single ALS and control line with different colors. Now, in the age of high throughput sequencing, we can actually barcode each of the lines with different unique DNA barcode, and that will allow us to mix maybe 50 control and 50 ALS lines together in single well, expose them again to differentiate into motor neurons, expose them to stressors, and then just by sequencing, basically find out how many of the ALS and control lines are surviving under different conditions. And I just wanted to point out that very similar approach was presented here at ISSCR, but Jana Mitchell from MIT and Broad Institute. Thank you.
Yeah, hi, my name's Ed Stanley. I'm from the Murdoch Children's Research Institute here in Melbourne, and I've worked with collaborator Andrew Elefanti on pluripotent stem cell differentiation systems for the last 2 decades, starting with mouse and then moving on to human. Our interests have mainly been around the blood system, and the blood is an interesting system in terms of disease modelling for a few different reasons. A lot of the common conditions that people see in clinics are related to issues with the blood system, and this can range from things like allergies to asthma, very common conditions, to type 1 diabetes, and more specific diseases like leukemia, which involves specific genetic changes within the blood cells. The blood system has a number of advantages as a experimental to try and probe human disease. One is it's very well characterised in terms of the way it's set together. The other great system it has is that it's possible to recreate a human blood system in an animal model. And that kind of experiment has been done over the last 30 or 40 years and is quite a reproducible procedure where you can essentially recreate a human blood system in a mouse. So the 2 things that I'd highlight today that we're interested in are the diseases that are, rather than being caused by a specific monogenic mutation, often involve potentially multiple loci, complex combinations of mutations, and a lot of unknowns. We have work that is going on looking at type 1 diabetes, and as yet the cause of type 1 diabetes is not well understood, but we do know, or at least suspect, that the immune system is involved in that. And so one of the kinds of things that we're doing is generating immune cells from individuals with type 1 diabetes and examining the properties of those cells. And this is something we can do both in vitro, but it's also something we can do by transplanting those cells into animals and allowing them to interact with other cells of the blood system. The other area that we're putting a lot of effort into now is in leukemia modelling. So you may or may not be aware that the current hypothesis about the way leukemias and cancers in general develop is that there are initially a set of priming mutations or driver mutations that set a cell up on a course to becoming a leukemia or a malignant cell type. However, by the time someone presents at clinic, those cells or leukemic cells may contain many different mutations, And the contribution of those mutations to the overall phenotype is very difficult to dissect. There are models that you can use to study that system. For example, you can take those leukemic cells and transplant them into animals and establish a xenograft model. One of the difficulties with that system, though, is that you're starting with the end product of the leukemia, carrying multiple clones with multiple different genetic alterations. So the question that we're using our pluripotent stem cells for is if we take each of those different specific genetic alterations that are mixed up in the final leukemia and separate them out into a system where we can inducibly control them, we can start to look at what are the initial events that trip a cell into becoming a leukemic cell. And so, for example, there are common mutations in pediatric AML or ALL, and usually by the time kids are treated, those mutations have a number of friends that have come along for the ride that have helped to transform that initial priming mutation into something that leads to a leukemia. So what we've done is we've genetically engineered inducible versions of those mutations into our pluripotent stem cells, and then using in vitro differentiation protocols, we're able to generate blood cells that are ostensibly normal. Taking those differentiated blood cells, we're then able to switch the oncogenes on and look at what happens immediately as those oncogenes become activated. And what we're hoping this will enable us to do is to, in a very reductionist way, deconstruct the initial events that happen in the genesis of a leukemia. And if we're clever enough, we'll be able to use those same cells as a screening tool to look for drug interventions that can either arrest or interfere in that initial priming event. I think that's probably all I want to talk about, so I'll hand it over to you, Joe.
So my name is Joe Wu. I'm a cardiologist. I'm at Stanford University. And I just want to emphasize, just like what Ricardo mentioned, it's very difficult for us to get heart cells from patients. So having the ability to generate heart cells from the iPS cells allows us to study the fundamental mechanisms of heart disease. And so in our lab and also in many other people's lab, we use these iPS cell-derived heart cells to understand disease mechanism. For example, the disease mechanism related to hypertrophic cardiomyopathy, dilated cardiomyopathy, cardiac arrhythmias, and so forth. You can also use these cells to do it for drug discovery or drug screening to see whether a drug is safe or not, especially in the area of cardio-oncology. A lot of times when a patient gets some type of cancer, the patients get chemotherapy, and these chemotherapy have quite a bit of toxicity on the heart muscles. And so we're in the process of using these cells to screen for the efficacy as well as the safety of these drugs. You can also use these cells for a clinical trial in a dish model, meaning that if you're able to expand these cells, instead of doing on 2 patients, 3 patients, expand it on 1,000 patients, 2,000 patients, and have a big pharma use their drug and test it on 2,000 patients at a time. And then based on which drug works and which drug does not work, based on these cells in a dish, then go to a real clinical trial. And then you can also think about using this, as Ed alluded to earlier, for cell transplantation, taking these iPSL, ESL-derived cardiomyocyte, inject into patients and see what happens. The last area is this area of this precision medicine, in which a lot of people want to use these cells to help them predict what's going to happen to the patients. So I just want to highlight one example here. And this is a initiative that was started by our previous president, President Obama, on the precision medicine initiative, in which the goal is trying to deliver the right treatment at the right time, every time to the right person. This is actually will be widely implemented by many companies. This is actually a press release by Geisinger, which is one of the largest healthcare systems in the U.S., in which they're planning on routinely DNA sequencing their patients. In the pilot program would involve 1,000 patients at DNA sequencing. However, this is actually not, there's some controversies involved in this. I think the public who's watching this video may be aware that there's issues with just routinely DNA sequencing patients, because for many diseases, especially heart disease, it's a polygenic disease. It's also, for many diseases, the DNA sequencing is a probabilistic, it's not a deterministic data. And so the public just need to be aware of this. And also, when you do DNA sequencing, many times, more often than not, it comes back as what we call a variant of uncertain significance. So you could get your DNA sequence, and the data comes back as, we just don't know what the DNA sequencing data mean. So this is going to be an issue if you're thinking about routinely DNA sequencing patients. So I just want to highlight one example. This is a case that's referred to us, in which you have a healthy middle-aged man who decided to get DNA sequencing for quote-unquote routine checkup. And the person turns out to have a pathogenic, it's annotated as a pathogenic. Pathogenic just means very likely to cause a disease, a pathogenic variant that could cause a hypertrophic cardiomyopathy. This is a thick muscle. The patient could die all of a sudden from cardiac arrhythmia. A patient could die long-term from heart failure issues. The problem is the patient himself is completely asymptomatic. The mother is also asymptomatic. The brother is asymptomatic, and the daughter is asymptomatic. So as a clinician, how do you advise the patient? Well, you could say that, don't worry about it. We're just going to follow you. But what happens if the patient passes away from some other unknown cause, and the coroner's office was not able to figure out what the cause is? And this is probably going to be subject to lawsuit because you have a DNA sequencing information that shows pathogenic variant that causes cardiomyopathy. You could say, okay, we're going to follow you very vigilantly, but in that case, if the patient happens to be somebody who's the worried type, anytime when he gets a skip beat, he's going to call the doctor and say, hey, doc, I have a skip beat. Can you scan my heart to see what's wrong with me? And think about the enormous cost of healthcare that's going to be related to this. So I just want to highlight one example of what you can now do with these iPS cells in genome editing, in which in this case here, you can make the iPS cells from the patient and the family. In this case here, we're able to show that the iPS cells are normal. There's no arrhythmia. There's no hypertrophy. We could also genome edit to make a double copy of this variant. And even with the double copy, we saw normal phenotype. You could also genome edit it so that you knock off the normal allele, so you're only left with the supposedly pathogenic allele. But again, in that case, it's also a normal phenotype. And lastly, just making sure that there's nothing wrong with the genome editing approach. You could genome edit so that with a well-known pathogenic variant that we know can cause a hypertrophic phenotype. In that case, we saw an abnormal phenotype. So based on these processes, we're able to tell the patient that what's originally called as a pathogenic variant is most likely a benign variant. And so you'll probably see more and more examples of this in the next few years, in which researchers and maybe later on the clinicians who are more aware of this particular approach will combine iPS cells and the genome editing to decipher what this variant of significance means for our patients. Thank you very much.
Great. So thank you very much. So let's consider a few questions. Question from the audience.
That's really interesting, the stuff about the genetic testing. I'm wondering what the implications are. Oh, do I need it? Oh, okay. I'm wondering what the implications are, particularly in the US, from a health insurance perspective, if insurers are now offering these tests and then presumably demanding them of potential customers, but we still don't know if these are actually going to be pathological genotypes. What does that mean for health insurance down the line?
I think the short answer is we don't know, meaning that a lot of times it depends on how much the firewall is. A lot of times you could get DNA sequence, and the provider will basically say this is for research application purpose only, and we're not obligated to tell you what the clinical information is. The other approach would be for the researchers to pass the quote-unquote hot potato to the clinicians and say that, okay, we're going to sequence you, and you have to talk with your geneticist, or you have to talk with your clinician to figure out what the clinical data mean. And so there's a gray area in terms of how do we, what do we do with these set of data? And I give you one real case example: a patient who decides to get DNA sequence, either for fun or for curiosity, and it comes back as tons of EUS that you don't know what to do with it. And in this case here, there's, as I mentioned, there's issues with how much to tell the patients. There's also issues in terms of, think about the psychosocial impact, the anxiety that the patient and the patient's family could have, knowing that they have some kind of variant that nobody seems to know what's going on. And so, I just showed this case because we think right now, from a research perspective, we could probably use the iPS cells, well-phenotype iPS cell cardiomyocytes, and the genome editing approach to help our clinicians with some of these difficult to interpret cases. Yeah.
And you mentioned that this is an in-court case, or you're predicting that it's going to be an in-court case. I'm presuming there's a dispute over whether the health insurer will pay for cost of treatment, or did I get that wrong?
No, so this case here, the patient just pays out of pocket to do the DNA sequencing, and so it's not covered by insurance. But I think the case I mentioned to you, Geisinger, they're planning on DNA sequencing 1,000 of their patients as a pilot study. Some insurance companies are very aggressive. My hunch is that some of them probably want to collect large amount of these data and for their own purpose, for advertisement, for commercialization, perhaps. So it just depends on which insurance companies is offering this to their customers. Yeah.
Questions. Sorry, I'm just going to dominate everything. So this idea of developing these patient-specific, whether it's an organoid or a culture in a dish, to test for drug discovery or whatever. I mean, how cost-effective is that? I mean, I realise it's fairly early days for this, but are we looking at a reasonable scenario of any patient comes in with a disease of some kind, they get sequenced, you generate the line with their particular mutation profile, you test which drug is going to be best for them. Is that a viable scenario for the future, or is there always going to be limitations in terms of the cost effectiveness of taking that individual level approach to disease management?
Well, I can answer that question, then maybe I'll ask Joe again, because I think he's the one who's been thinking most carefully about iPS-derived models as diagnostics. I think as it stands today, it's probably too variable and too expensive using the technology that we have. But of course, the technology can improve quickly. And we have shown that we can automate the process. And so that suggests that one might be able to do it more routinely. I think that there's still issues of reproducibility and issues of identifying the right phenotype for specific diseases that make this fairly impractical today. So, I mean, I think in principle, for example, you'd like to be able to determine which antipsychotic to give somebody or which antidepressant, and it would be wonderful if you could do that by taking some of their neurons and testing the responses to different drugs. That's not completely viable at the moment at the price point for a large number of people. But Joe, I know you've been thinking a lot about this.
Ed was going to chime in.
I just say that you have to remember that the knowledge base is not going to be static. So as people get more and more sequences correlating with more and more conditions, the need to make models is going to become less and less. And so you could imagine in 5 years' time that the kinds of things we might be looking for in terms of modelling will not be the kind of things we're doing now, because there'll be just so much more information out there. That's the only thing I'd say.
Yeah. And I think to follow up on what Ricardo was saying, maybe 5 years ago when we put in an NIH grant, we put down $10,000 to make a line. And right now, when I put it in, I put down $2,500 to make a line. So over time, the cost has dropped significantly. And also in the area of cardiac, 10, 15 years ago, we're only getting about 5% differentiation. 10, 15 years later, we're getting 90, 95% pure cardiomyocyte. So as more and more people work on it, the field is going to improve by itself, and so you'll see the cost goes down, efficiency goes up, specificity goes up, and hopefully the maturation of these cells will go up. I think the main point I also want to emphasize is that at the end of the day, this is not alchemy. We're not trying to take sand and turn it into gold. This is real biology behind it, because for practical purposes, these iPS cells are the same as ES cells. And for the ES cells, it becomes you and me, and the ES cells become all the different organs, different tissues, different cells. And for the iPS cells, you should be able to do that. Right now, we're just not smart enough to figure it out in 2018. But in 10 years, 15 years, I think you'll see tremendous amount advances in each one of the areas. Yeah.
And I just had a question for Dr. Wichterle. With the work that you do with organoids, and you talked about a lot of the variability in terms of not only over time, but also the type of pathology and other factors that enter in. How do you work with the data sets there if there is so much variability, or is there a way to have standardization in any way that can be used more broadly?
Yeah, so we try, of course, to minimize the noise in the conditions and in the cell lines. We are also using, because during the differentiation you don't end up with one type of cell, so we also introduce reporters in the pluripotent stem cells, so we can follow only very specific cell types in the dish, like, for example, motor neurons. And so by doing all of these sort of careful manipulations, we can reduce to a large extent the noisiness of the cultures, but it's not, we are not eliminating it. And so I feel that specifically in these degenerative diseases where the phenotypes are so subtle, there is a maybe unconscious tendency to pick lines, disease lines and control lines that behave in certain ways in laboratories. And I think that the only way to avoid this issue will be, in the end, to develop these multiplexing approaches where you can mix many different cell lines and reduce the noisiness in this way, basically increase the significance of the small phenotypes that you observe.
Me again. So you're sort of operating in fairly organ-specific areas, but I guess particularly with metabolic disease, there seems to be a multi-organ issue. How is that likely to work when you are working on such organ-specific cells or cell lines or organoids? Is there a way of, is that the kind of the next step of actually looking at this in a multi-organ perspective?
Well, I would say that in a sense, sort of diseases and mutations that affect multiple organs, like, for example, mitochondrial diseases, have some advantages because they are attacking some aspect that is shared among all cells. And so, in principle, you can select a cell type that is relatively easy to work with and try and understand the disease in that cell type. And then you can do sort of validation studies in different kinds of cells. And that is sort of the approach we've taken when we're trying to deal with these diseases that have both a CNS component but have, components in the periphery as well. For example, lysosomal storage diseases, right, where we have treatments today that work for the peripheral symptoms but not treatments that work for the CNS. But that's largely because the treatments don't get into the brain. So the underlying phenotype is similar, and the underlying problem is similar. So I guess what I would say is this: you don't have to build a whole human. You can build one cell type and then use that to test things in other cells, and then ultimately do the experiment in the really only experimental organism that matters, which is humans.
Sorry, I promise it's my last one. One of you mentioned that you're now using these cell cultures as a kind of a way to filter, sort of preclinical filtering for looking for drugs that might be likely to work and then take into human trials. How big an impact is all of this work going to make in terms of reducing animal studies and reducing the need for animal models? I mean, is it likely to do away with it entirely, or is there always going to be a need for an animal model at some point in clinical research?
So, I think that in our case, we can test our drugs side by side in human motor neurons and also in mouse motor neurons. So we know which drugs are effective for both species and which drugs are preferentially effective for human but not mouse. And in my view is that since the eventual clinical trials in patients are really very costly, if there is a drug that we see efficacy in mouse, I would not hesitate to go first through the mouse model and see whether we get, again, any indication of efficacy in there.
Yeah, the other thing is that some drugs require to be processed before they either work or might have side effects. And so even though a drug might have either positive or negative effect on a particular cell type, like a motor neuron, it's possible that once a living creature got hold of that drug, it might go to the liver where it gets turned into something good or bad, depending on which way you want it to go. So I think there's probably going to be still a requirement to go through an animal where the drug can display all of its potential characteristics. You would be difficult at this stage to model in vitro.
Especially toxicity studies will have to still be performed in animals because we are testing in one cell type or a few cell types in a dish. That's not the same as testing it in a living organism.
Yes, so I think you need both. I think traditionally the drug discovery process has been, you have compound library, you come up with hits. With these hits, you put in animal model, typically a mouse model. And depending on what disease or cardiovascular, a lot of times you have to go to a large animal. For cancer, you probably don't, and then go to human studies. I think my beef on the animal model is that traditionally the drug company, for example, they will pick a particular strain of mouse. They could pick, for example, a FVB mouse strain, and then they will show that the drug works on 500 mice here, and then 500 mice with placebo did not have a positive effect, and therefore the drug works. In essence, when you think about it, because these mice are all syngeneic, meaning they are inbred, essentially you've shown that the drug worked for one person. You've shown that the drug worked for 500 of my clones. And so what the iPSL diversity brings in is that, if you're able to reverse the model a little bit so that you have the drug and you test it on 500 patients with the drug and the 500 patients without the drug, you're able to show that this particular drug works quite well, and you're able to further delineate and show that maybe for some reason it works quite well for Caucasians, Hispanics, Asians, but not African Americans, and you're able to tease out the molecular signal. And then you go to the animal model to show that there's a safe PK/PD, pharmacokinetics, pharmacodynamics, and then you go to a clinical trial. So I've always joked, not joking, but in seriousness to the drug company that if they have a drug, don't show me the drug works in FVB mouse. Show me the drugs work in the mice that are running the streets in Melbourne. And if they work, then I believe the data. If they don't work, then I don't believe the data, because the clinical trial is much more heterogeneous compared to animal studies. Yeah.
Yeah, I would also add to that that I think the thing that has changed really is that we're sometimes willing to go forward with things that just work in the cells. So we're willing to use the cells as an efficacy model. We're still not at the point where we're willing to use cells in a dish either for pharmacokinetics, that is to say how the drug is metabolized, or safety. But there are, in fact, in neuroscience, very few really good mouse models. The vast majority of the models are not very good. And so we have this long history of failure, mostly because we spent all of our time optimizing drugs for things that made, mouse swim better in a bucket, or, mouse make decisions about whether they want to stand on a beam or not. And it turned out that those assays were not very predictive. And then when we look at genetic models, it turns out that many of the genetic diseases in human are loss of a single allele of a gene, whereas that are really well tolerated by mice. Mice tolerate just loss of one allele really well, so they're not a very good disease model. I would say it's actually the exception that is, it's the unusual mouse that is a good disease model. Most mice in neuroscience are really pretty terrible. So for many of our programs, we've sort of said, well, look, we believe in genetics, and we believe that the cells in the dish are more or less conserved, and if we can fix whatever the defect is, because we think that that's good enough for efficacy, and then we will do all of the other studies to make sure that it's safe and that it gets to the right place and that it engages the target. And that actually has the sort of unexpected side effect that it makes the whole process much faster, because these animal studies take thousands of mice and take really long periods of time, whereas doing it in cells is much faster. So
I had a question about the DNA sequencing. Basically on the data that is gathered through the processes of sequencing patients, for example, through all of the Geisinger patients, is that data set useful for any kind of crunching to determine what some kind of screening process in the future? Is it useful for other purposes?
So, go ahead, Joe. Well, so I mean, many drug companies are trying to take advantage of the combination of large-scale DNA sequencing and electronic medical records to try to understand whether the drugs that we use today have interesting and or deleterious side effects, and whether that correlates with specific variants. So far, we don't have large enough groups of patients. People are too diverse, especially when you look at common variants. But there are a few examples where we have learned from the combination of the DNA sequencing and the electronic medical records that there are some mechanisms that have completely unexpected effects that means that they might be useful for other disease indications. And an example of this is the development of ketamine as a fast-acting antidepressant. And it really came about because people started looking at all the people who were using this very common anesthetic for pain, and they found that only a small fraction of, that the people who were using it for pain were not actually depressed. They had a much lower rate of depression than people who normally have pain, and that this was associated with specific genetic variants. And so this has then led to the development of ketamine-like drugs as fast-acting antidepressants. So this is one example, but Joe.
No, I mean, I think Ricardo said it right on the spot. I think the other main issue is what do we do with the data, meaning that you get your DNA sequence as a consumer, who owns the data, and whoever who sequenced you, what they can do with the data. And that gets into this medical legal gray area zone. It's almost like the Facebook with the Cambridge Analytica issue. So that's something that we have to be watch out for in the future in terms of DNA sequencing. Yeah.
I mean, I'd just chime in again and just remind people that things progress very quickly, and it would not be surprising in 10 or 20 years that everyone has their DNA sequence done and they carry it on their iPhone and they're in control of it. So I think that at the moment there's a window where it's too expensive for it to be part of your personalised Facebook page, but it would not be surprising in the future if everyone kind of knew something about their DNA sequence, and that might happen faster than us old people would appreciate.
And I would just highlight that in the case of ALS, this is also very important because only less than 10% of ALS cases are familial or they are connected with a single gene mutation. So despite this very low number of familial cases. At Columbia University, we now sequence pretty much every single ALS patient because we hope that by collecting large enough data set, we will be able to identify perhaps even modifiers of the disease that would not be fully penetrant but would affect when the disease is manifested or how severe it is. So I feel that by building the database, we are really building a very important resource, even going into the future.
So Hynek, one of the issues that has come up over and over has been this question of how well the cells that we make in the dish actually recapitulate the disease, particularly because they're very immature. So what sort of progress are we making in trying to improve the maturation of the cells? Because at the moment the cells mature more or less at the rate of humans.
Yes.
And so what is your approach to that?
So this is, I think this is really the crux of the problem of dealing with late onset diseases. And I think the initial approach was that we just grow them in crappy cell culture conditions, and they're on their own stressful in some kind of random way, and that might mimic some kind of stresses that are associated with aging. I would say the second permutation of these studies was what I described, where we tried to actually find very specific stressors that would actually activate pathological pathways in the cell that might then synergize with the mutations causing these synthetic lethal phenotypes. But I would say that in the future, it's going to be really much more faithful recapitulation of the aging process. Unfortunately, most of neuroscientists study the specification part, how you make different types of neurons, but there are very few studies that actually look at what happens once the neuronal identity is specified, and this cell has to live for decades in patient. What are the genetic changes, molecular changes that are associated with this aging process? So my lab now started to work on this question, and the first, basically, maybe a little bit reductionistic approach is that we think that there is going to be transcription factor controlled program that will control the maturation process. And if we can identify what transcription factors control the maturation, we will be able to use them to reprogram the embryonic cells into this mature state. And I feel that we already showed that you can do it during specification steps, so it should be feasible in this maturation process as well.
Yeah, no, I think that's a really exciting idea. Joe, how about you? How are you? You're working on heart failure. Again, this is something that emerges later in life. How do you model this?
It's a difficult issue with the iPS cell cardiomyocyte maturation, and I think, just like any other fields, the iPS cell derivatives, brain cells, liver cells, kidney cells, heart cells, endothelial cells, they're more of an immature phenotype. I think in our field, people have tried different ways. So, for example, make it into a tissue engineering construct and try to mature the cells that way, try to paste the cells, and try to add different kind of metabolic substrate to mature the cells. But I think, as you pointed out, at the end of the day, the human heart cells have gone through 20, 30, 40 years of beating. The heart has been beating and undergone 20, 30, 40 years of different kind of environmental stress, whereas the cells that we make, we're not subjecting them to that. So I think at this moment, it's a difficult process to mature them. But I think perhaps with more people working on it, people will figure out either turning on some kind of transcriptional factors or stressing them in shear stress or different kind of conditions to make them more mature. But at the end of the day, I don't think, my hunch is that the cells that we make will never be the same as adult cardiomyocytes, adult human cell. We can get it much more similar, but you'll never be the same. And the main reason is, for example, conversely, if you take a liver biopsy from a patient and put the liver on a dish, the first day it looks like your liver, but after 7 days, the liver cells have kind of de-differentiated because on a dish condition, it's not the same as a human body condition. So you can get something similar to adult cardiomyocyte, but I don't think it'll ever be the same. Yeah.
So, thank you. So a lot of people have been using CRISPR to engineer stem cells to correct mutations, to generate mutations in a nicogenic background. But recently we've learned that CRISPR-engineered cells can develop second mutations because they respond to double-stranded breaks by undergoing apoptosis. And so the cells that are actually mutated often have second-side mutations in p53 and in other genes. So so Ed, you've made some of these mutations. How do you, I mean, do you think, first of all, that that's an issue that affects your modeling? Is this something we should be concerned about? Is it just something that emerges in, some unusual cell types?
I think that from a modelling point of view, it's not something we worry about, simply because the experiments that you do need to be set up in a way that you might use multiple lines or conditional systems that allow you to isolate the variable you're looking at. Obviously, the point where that will be an issue is if you're trying to make cells that you'll want to use for transplantation.
Yeah. Joe, how about you? You've also been using CRISPR to correct mutations.
Yes, so I agree with that. I think probably for the cell modeling, for the disease modeling, probably not much of an issue. And it also depends on where the mutation is. If the mutation is a very dominant oncogenic mutation, then you probably don't want to use that particular line because it's going to be highly proliferative and it'll probably be different from some of the quote-unquote normal iPSC cardiomyocytes that you have. So for drug screening, disease modeling, probably less of an effect for if you're thinking about cell therapy, that's probably a much bigger issue. Yeah.
But I would again just point out to the fact that these mutations often make embryonic stem cells or iPS cells divide faster, and so eventually they often become the favorite cell lines in the lab because people like lines that grow faster. Yeah. So I think it is an issue even for modeling, and again I think it highlights that we will need to have multiple lines in parallel where we will compare the behavior.
And then, along the same line, it doesn't have to be CRISPR gene editing, right? The fact that for these cells, you're trypsinizing, replating, that's a very harsh condition. Humans don't go do trypsinization and replating, right? It's a harsh condition for the cells, and over time, you'll have cells that have acquired mutations just because of that process, independent of genome editing.
Yep. Okay, well, thank you very much. Thank you all, and thanks so much for inviting us.
Thank you.
Thank you.